Welcome, my fellow classmates. Today we are doing our project for our Foundations Science class, Understanding DNA. I'm Justin, and my partner in this is Katie. We've prepared this short instructional video for your benefit and hope that you find it fun and informative, but we do apologize that we are not professional videographers. I first want to start off by asking the question, what is DNA? DNA is a nucleic acid that contains the genetic instructions used in the development and functioning of all known living organisms. So what is the function of DNA? DNA serves in all cells of living things. DNA is what builds you and me. The role is the long-term storage of information. DNA can also be likened to a set of blueprints. These are the instructions to build your cells, such as protein and RNA molecules. Let's go ahead and break down DNA. DNA is made up of three groups: a phosphate group, a sugar group, and a nitrogenous base group. To start off, phosphate/sugar groups are the same for all nucleotides. As we can see, Katie is pointing to the groups, and it's important to note here that a phosphate always connects to the sugar, and looking at this DNA strand, it's on both sides. Now, to get to the bases are different between each nucleotide. There are 4 bases, and those bases are known as thymine, adenine, cytosine, and guanine. An important thing to note here is that with thymine and adenine, they always bond together. And the same goes for guanine and cytosine. Those base pairs always bond together. You will never ever see an adenine bind or bond with a guanine or cytosine. And the same goes for you'll never see a guanine bond with a thymine or a cytosine. An important thing also to mention here is that the phosphate is connected by what is known as a covalent bond. A covalent bond is also known as the strong bond and are very difficult to break. That is why it is known as the backbone. The base pairs are connected by what is known as a hydrogen bond, also known as the weaker bond. These are easier to break than the covalent or strong bonds. Adenine and thymine, or A and T, are connected by 2 hydrogen bonds, whereas guanine and cytosine, or G and C, are connected by 3 hydrogen bonds. Those hydrogen bonds, the differences in those hydrogen bonds makes A and T easier to break apart than G and C. And that's important to know because it comes up in a later function of DNA known as replication that we will cover. One DNA strand is completed by linking different nucleotides together, again making up the backbone of the DNA. And another thing to note is that it is in a repeating pattern. Another thing to note here is we have the DNA strand laid out in ladder form. DNA is actually in the shape of a double helix, but for simplicity purposes we have it laid out as the ladder. A very interesting and quite amazing fact about DNA is that if DNA in a single human cell were stretched out and laid end to end, it would measure approximately 6.5 feet. Wow, that is big. The average human body actually contains 10 to 20 billion miles of DNA distributed among trillions of cells. Now, a question might be brought up of how is it possible that there's that much DNA strands in the human body? Well, as we have mentioned, DNA pretty much makes up our life. It, in a sense, kind of defines us. And so it's important to have many copies or duplications of the DNA to make who we are. The process by duplicating or multiplying the DNA is known as replication. There are 8 things that are important to know about replication. The first is replication actually happens just before cell division. Number 2, to replicate, the double helix must be split first. Number 3, replication starts at sites along the chromosome called origins of replication. Very original, right? The origin is actually a specific sequence in the DNA that is recognized by DNA polymerase as a start point for replication. Number 4, each strand is actually a template for building a new strand. That actually brings us to point number 5, is that DNA polymerase binds together new strands and cannot make a new strand by itself. Replication can only occur in one direction. Number 7, again as I mentioned, DNA polymerase can't start a new strand of DNA by itself. It needs to have the template from the original strand. And the last important thing to note is number 8. Once a new strand is created, it is proofread for mistakes. 2 points in that is that before editing, the error rate is 1 in 100,000. But after editing, the error rate is actually 1 in 100 million. So it's very small and uncommon to find errors. But if an error is found, errors can be passed on and create mutations from one generation to the next. So in DNA is actually really the ideal material for carrying genetic information because it's able to store a vast amount of information. And that information is then to be translated into physical characteristics. DNA can be copied quickly and accurately and is passed from one generation to the next. Or in other words, it is heritable. In concluding, we hope that you really understand the fact that DNA is important to life. DNA is the building blocks of life. It's in everything. It defines who we are, and it's what makes every single person different and unique, all the way from hair color, eye color, to size and stature. Well, that is our video of DNA, what it is, and the parts and its function. We hope you enjoyed this video and found it very informative. Thank you, and have a good day.